So, okay, the session is recording. So this is the progress that we have so far for the OmicsLogic Data Science program. And I see that a lot of you have been doing tutorials on the Code OmicsLogic platform, which is clearly denoted by the pink graph that we see here. So if you are to do more courses on the EduT Bioinfo platform, then it would be denoted by the yellow. While if you are to run pipelines on the server platform, it would be denoted by gray. So if we come down here, we can look at individual points for the different courses you are doing. So I see that most of you have not yet started with the courses that are there. So I would encourage you all to go ahead and take the courses. While I see that we have been going over the lessons in R and Python a lot in these last 3 sessions that we have. So we are seeing some progress on all of your part in the Code OmicsLogic platform, so which is good. Now I encourage you all to go through the courses which are also listed here, as it is also imperative that we learn both from the things that are there on Code OmicsLogic as well as all the courses that are also listed down here, especially starting from these basic introductory courses, which is Introduction to Bioinformatics, Introduction to Genomics, at least these 2 introductory courses. And then we can go ahead and start taking up more advanced courses like Genomics 1 or Transcriptomics 1. And if you have any questions, then please put those questions right here in the forum page. And it will also help us reach out to you whenever you have a question. So you necessarily do not need to email us. You can put it on the forum and any of our mentors would be able to reach out to you and answer those questions whenever you are posting them. So it's an easy way. And also I'm getting questions regarding where can I find recording for all the sessions. I'm not seeing the emails. So if you are to find the recordings of all the sessions posted here, you can find it right under the session recordings. So this will actually show you the different sessions and the recordings, and I keep on updating this every day. So you'll see all the 3 sessions are posted here. In case you have any questions again, or you want to post something related to the sessions, you can also type it out here. You can type out any of the other technical problems that you would be facing right here. And you can also see that Dr. Harpreet has posted about some technical sources and data sets, which I would also again encourage you all to go through them and post your own feedback, review, or your questions right here in the forum. So you can see the data set that she had posted. Now, I would like to pass today's session on to Ilya. Ilya, would you want to go ahead? I'll stop my screen sharing now.
Thank you, Bipsa. So in fact, today we're going to change the way that we've been doing things just a little bit. We'll start off with a review of what we did last time with Dr. Harpreet Kaur, and she'll kind of go through briefly in just a few minutes some of the things that we covered, and then we'll pass it on to Clinton, who will talk about an introduction to Python and a review of visualization in application to the gene expression data that we started talking about last time. So Harpreet, would you like to?
Yeah, thank you, Ilya. So first of all, I would like to ask, many of the, some of the problems that you are facing. So let me ask first Sylvia. So Sylvia, are you able to install R on your system?
Hey, good morning. Can you hear me?
Good morning. Yes,
oh,
you are
okay,
audible.
Excellent. Well, thank you very much, because I could install R version 4.03 and also the last version of RStudio. Thank you.
Okay. So next,
but I have a problem with, I have a question.
Mhm.
Happy, which is when you use a skip equal 1,
Sorry,
I can write it on the chat.
row names equal to 1. We are keeping
Yeah,
that.
no.
Oh,
She's asking about skipping the first row. So when you have your numeric data and you have an additional row, for example, you can specify the header, right? And the header will be the first row. But then you also have the group names. And if those group names are there in another row, then you need to skip that row. And so that's when we use that option in the loading of the data.
and we need to remove that row. The reason is that that is non-numeric. They it contain characters. So because for the visualization purpose, eventually all the plots going to take your numeric data. So that's why we are actually removing that line. That is first row. That is header. Is it clear to you now, Sylvia?
I understood more or less. I have problems with running a code um, on the lesson 2.
Data visualization. Okay, so
Yes.
let okay, so let
The first script, I can't run it.
okay. So let me just go over there. Then you can let me know exactly what issue that you are facing.
Yes, the first one. Yes.
Okay, so okay.
I don't know why I can run it. Yes, the result is okay. I can get
Okay.
the, but they say the you run it good, but that is not the right answer.
Okay, so can you try again?
But not in this moment because I could should and okay I could because it goes slow. My computer goes slow if I have to.
Okay. Okay, so okay, it's okay. You can run after the session as well, and then write me mail whether you able to run it or not. So, are you facing any some issue? So here we are doing only what we are going to actually in the first step. We are just reading our file, right? So first
Yes.
we with the read dot table we reading that file. Then we just removing the first row here case. Otherwise you can also define row names equal to 1. So that is the both they're the different ways how you can skip the first line, right? So then we just converting into the matrix, and this is just to check whether this data get loaded. So there are different ways with which you can check. So already I told you that we can check the dimensions with the dim command. We can also check top 10 rows with the head command. So this is just here we are computing the mean of whole that data. Although this is not like for some purpose here, the purpose of this particular command that we are actually checking whether our data get loaded or not. That is what we are doing in this step. So the another question that Lydia has, although I have tried to resolve, I try to address her. So Lydia, is it clear to you why this colors are actually what they representing? So you can type yes or no in the chat box as well. So although we are not using this particular command in or in this code, but the let me just again address this what it representing. There you can, if you suppose you want to give multiple colors to your samples or the genes, so you can actually define colors for each of your sample. So this is these are just a function. So like wrap is actually a function, then we with the C actually this is kind of total vector. We are defining different color depending upon how many samples are there. So if you have, so it can be useful to the samples in different colors. Yes. The other way we actually also define, I mean, color the sample based on some color, I mean, their name or ID or group. So this is if you don't know sample belong to which class. So you can actually, depending upon how many samples are there, so you can define color for each of these sample or gene as well. So this is a purpose of this is just how you can actually define colors. Although we have, I think here we have 15 gene. So you can if I look at this data, let me look at this data, and we can also here define row names equal to 1. Then we can check with a simple, if you want to check how many rows and columns are there, simple command is dimension command. So we can check with that is, first it representing 15. So the first part is actually represent the number of rows. The second part represent the number of the column. So in the rows we have genes. We can look at even this data by doing head. So in the rows we have genes, and in the columns we have samples. So you can actually define, if you want to define based on your color, to whether you want to color the gene or whether you want to color the your sample, as there are large number of samples, that is 52. So sometimes it would be difficult to color each of these samples. So what's the way? This is one way. If you have, like, a reasonable number of samples, like if you have 5 sample, 10 sample, you can define color for each group. And this number actually, after the color, it is not representing the number, I mean exact sample number. It represent uh, that is actually so actually there are even color have different shades. So based on different shades we actually define these. If you have more number of samples, there are only 7 colors. So to give different shades, you can actually give these different numbers. It will eventually results a different shade for each of your sample. So the last time we have covered the another, anyone else who are facing any other issue?
Harpreet, a question.
Yeah, please go ahead.
Going to the first script
Sorry.
of the, could you go to the first script of this lesson 2?
Yeah.
I changing the root. It is not necessary, or it is necessary. I can change the root to the gene expression table or not.
Mm-hmm. I mean, the only, okay, so sometimes, okay, so let me clear one more thing. When you run this code, it is not your, if you get this like success running code, but answer is correct. Sometimes you change this data, right? If you're changing the data, or you, although your code runs successfully, answer is incorrect because we have actually defined already, I mean, what is the mean of, for example, here we had this particular, this particular code, and here we are computing mean with the df. So we have already provided that answer. If you just running this code and getting this answer, so you should get the same answer.
Harpreet,
So mm-hmm.
excuse me, I understood that the root had to be changed to the gene expression table, so it is not necessary to change the
She's talking about the URL. She's talking about the path.
Okay, path.
Okay.
Path. Okay.
Yes.
Okay.
Yes.
Excuse me. Thank you.
Okay. So this.
I don't require to change the path.
can change that. I mean, if you have, I mean, providing some, if you have data at some, you know, repository like GitHub or something, then you can define that path. If
Excuse,
you're running that
excuse
script
me,
on your own system, in that case, you provide the path of where your input file is located.
Harpreet, and I don't understand why you include this gene expression table.
Mm-hmm.
Which is the reason you give us the path for gene expression table.
So the way this exercise works is it shows you how to load data. This data could be any type of data. It could be survey data. It could be a small table of expression, or it can be a big table of expression. So what Harpreet asked you to do was to visualize a particular gene from the gene expression table. The reason for that is because we also now are covering transcriptomic data analysis. So you should be able to understand not only how to work with intuitive data like surveys, but also genes.
So I don't, I can use the same code you have in this first script. Don't require to change anything.
You're not required. It will still work,
Okay.
but you can also change it.
I
Yes.
was working for 1 hour to try to do this, so I don't require to change. Oh, thank you very much.
Yeah.
So now I would like pass this session to Clinton, who is going to take this session, and he is going to talk about introduction to the Python and exploratory data analysis. So Clinton,
Hi.
are you? Yeah.
Yep,
So
I'm
over
there.
to you, Clinton.
All right, awesome. So I'm just gonna share my screen
Yeah, go ahead.
and perfect. Can everyone see my PowerPoint? Yeah, just write a yes on the chat or something, whatever you want.
Yes, your screen is visible. Yeah. No, you can go ahead.
All right. So what we'll talk about some important subtopics within today's exploratory analysis portion. So we'll try to we'll review. Well, this is review obviously. So we'll review about the omics data types that we spoke about. We'll review about the formats and challenges. We'll review the pipeline. So how will you change raw data into a finished product to answer an analytical question? And then we'll talk about some big machine learning tools within this analysis. And then we'll review about finding a link between the data, biological data, and an actual question. So you need to interpret the data that you process. All right, so, oops, sorry. For some reason, my PowerPoint is missing that particular segment, but let's jump to this slide. So essentially what we'll talk about is some summary statistics. So we learned how to make histograms, box plots, and scatter plots, and what they represent. So they show the distribution. And can someone tell me what a scatter plot represents? Can someone write that in the chat? So if I were to create a scatter plot, what am I trying to visualize?
Different points around the graphics.
Yeah, yep. So essentially you're trying to compare two different types of data to see exactly a relationship. That's a nice word. Perfect. Similar patterns can be noticed with histograms and box plots. So you're trying to see the distribution of data. So whether the mean and the median are different from each other and how far apart they are. And in previous lessons, we also spoke about normalization of data. So sometimes it's hard to compare raw data to itself. So can someone tell me why it might be hard to compare raw data? Can someone write that or in the chat, or maybe even say it in the voice, or whatever is comfortable for you? So yeah, if I have raw data and I wanted to compare it immediately,
Well,
what are some challenges?
I could say that the raw data is not well organized, so we will not see any relationships or any pattern on the data.
Yes, exactly. You have, like, if the data is not organized, you can't really, you're comparing bananas to Lamborghinis. You know, you need to clean up your data into a proper format. And oh, Lydia mentioned gene can have a lot of reads or not. Usually it's in between genes. So yes, so sometimes you might have a lot of reads in one sample, but another one you might not have a lot. But is that a technical issue, or is there some actual biological significance behind that? So normalization is key when you have raw data, and you've already gone through log scale and quantile normalization, both very important within biology. And then, yeah, missing data, sometimes it's not good to have zeros and random. Oh yes, yeah, it depends on sequencing. Perfect, yeah. Sometimes, yeah, that's another application as well. So it's good to have, you learn this later on, but you can create graphs about depth and coverage, and that also gives you more insight on whether your data is fully representing the sequence that it is focusing on. So good point, Lydia. Perfect. All right, so we reviewed gene expression tables in one of the previous sessions. So can someone tell me what the columns represent and what the rows represent? You can either type it in the chat or say it in the audio. So if I gave you this data right now, yes, perfect. Which one's which? So genes and samples, but which one's which are the rows genes or are the columns genes? So usually the way it's organized, if you don't remember, is that for some reason labels aren't here, but I will annotate. Can someone give me host privileges so I can annotate? Anybody's there? Yes, rows are genes. Perfect. And the columns are samples. Perfect. That's great. So usually when you do a RNA-seq experiment, your end result will be an Excel or a table of this form, which you will use to process data, or which you need to process. Perfect. And it can come in fragments per kilobase million or transcripts per kilobase million. So here's just an example of what we learned previously about Excel bar charts. And essentially you can see, can someone tell me the difference between the left and the right? So what's the difference between both genes? Is there any difference? So what is the gene on the left? Like, can you compare the two? Which one, can someone give me some insight? Left is low express. Nice point, Lydia. Anybody else? Yep.
Do you see
So
the patterns of this gene being expressed on different samples
yeah,
as well?
yeah, exactly. You can see a pattern. Yes, perfect.
And
So
different levels of
yeah,
how much, and you can compare sample with sample, but also different genes at the same sample as well.
true. That's a very valid point, which is great. Which is exactly why we're making graphs like this, because we can see that there's a trend in the right graph that more samples express that gene. So maybe, I mean, I'm obviously guessing we might need more information, but maybe that gene is more of a housekeeping gene because it's regulated in most of the samples, while the left gene might have a specific function because it's in certain samples. So yeah, you can come up with important insights. And we also reviewed some nomenclature. So in Ensemble, they have specific nomenclature. So on the right, they don't mention the human. There's a 3-letter code, but on the right, sorry, there's a 3-letter code for mouse, but on the left, there's no 3-letter code. All right, perfect. And then genes and transcripts have different codes. So G is genes and T is transcript. So depending on the database that you use, gene can be on columns. Usually it can if you transpose the columns, and it depends on what you're trying to analyze. So normally they're on the rows, but it depends on how you're trying to analyze. So maybe if you're doing PCA later on, maybe you might want to put genes on your columns, but normally it's samples on top, but it depends on how you want to analyze it. Different genes expressed in mouse, human, and mouse. Yes, perfect. Yeah, true. Yeah, that is true. Sometimes, which one is the variable? Oh, for this particular, so the bottom is samples, and the top is the gene. The title is the gene, and the axis on the y-axis is the counts. So the gene expression values. So this particular sample had maybe 101 kilobase TPM or RP reads per kilobase million. So yeah, the x-axis is samples and the y-axis is the counts. Okay. I hope that answers your question, Sylvia. If not, you can ask it again, or you can just clarify. Yeah, so the variable we're analyzing is the gene. So we're trying to see the, yes, exactly. Yes, the counts of the gene. So yep. Perfect. All right. Now let's go to the next slide. So as you can see, we just reviewed some ways of plotting data previously. So on the left, you see a histogram of gene length versus frequency of a particular of the human genome project, particular data set. So you can see that there are certain genes over here that occur maybe 1,000 times with a gene length of maybe 1,000 here, and then up to 5,000 nucleotides. And over here, you can see on the right that this data has been log scaled or either quantile normalized, and it fits the correlation line. So usually when you see scatter plots like this, you want a high R-squared value because that means there's a better correlation there. So I hope you remember that from last class. And if you have questions as we go along, just type it in the chat and I'll try my best to answer them. So then we also reviewed how to create these box plots, and essentially these plots tell us whether important values such as the max value, whether they're outliers, the median, the interquartile range, the upper quartile, lower quartile, and these summary statistics help you in determining whether you want to remove outliers at an earlier point, or whether you actually see a trend in the data. Maybe you don't see anything and you might not want to pursue this particular data set. So these metrics are always important to do because this is all quality control. So you want to make sure you're not going down a rabbit hole or you're wasting your time downstream of your initial analysis. So can someone tell me what's wrong with this picture? Can someone write in the chat? In this example, conditions are very different. Yes, in this one, and this one, yes, perfect.
Is it that in the previous box
This one,
plot,
right? Yeah.
the other one? This is more. This is well defined. You can see the mean and the low and higher quartile.
You mean this one?
No,
the first one?
You asked about.
Sorry, my bad. Maybe I went too far back. Sorry, this one, right?
No, the other one. Were you talking about the other one, or you asked what is?
Oh, okay, my bad. Sorry, you're talking about this one? This one, right?
Yeah, is that the question?
My bad. Yeah, this is the question. My bad. I thought you were talking about the previous one. Sorry.
No, I was just trying to answer the question regarding this one. And here you're looking at the data. It's at the bottom of your scale. That means it's not well defined. You cannot see the mean or any.
Yes.
The box plot, actually, you see the top outliners.
Exactly. That's a very that's the exact point of this plot, that you can't really tell any differences. All you see is outliers, and that doesn't really tell us much. So this is where normalization comes in handy. So it will help us see the actual mean and push the outliers farther out. So good response. Perfect. See, majority of values and outliers. So as you can see, if you do log scale transformation, you notice that there's a change. You can actually see the median, you can see the interquartile range, you can see the min and max. But if you don't normalize it, you just see outliers. So it's good to normalize raw data, because you can actually come up with trends. So perfect. And and the same trend here. So can someone tell me, can you come up with a trend when you look at the graph on the left? Can you make any inference? You can say yes or no. It's not a hard question. All right, I'm not trying to make it hard. Can anyone tell if there's any difference? It's a count plot with so the genes on top, and yes. So it's a histogram, and it shows you the counts, and it shows you a particular gene over here. So yes, you mentioned that you need log, and perfect. That's why on the right, you know that you can actually see a trend. Yes, non-normalized data, perfect. Yep, non-normalized data, you can't really see a trend here. So in this histogram, you can see a trend after you normalize the data, which is exactly why you need to do quality control or exploratory data analysis. And the same thing with quantile normalization. This technique uses the means of different samples and arranges them so you can actually compare them. So it's another way of normalizing that you should have gone over. And it's perfect. Depending on the circumstance, you can either use log or quantile. So normally with gene expression, you can use either or. So yeah, and perfect. It's the same trend as early on. You notice that you can actually compare data more accurately when you quantile normalize. And sometimes when you do quantile normalization, you get rid of. Okay, let me ask you this question. What are the benefits of normalization, like quantile normalization? What will happen if you normalize? What will you get rid of? Can someone answer that question? So if I do quantile normalization, what am I getting rid of really? Okay, I'm getting a bell-shaped distribution. Okay, I'm getting a normalized distribution. That's perfect. But what am I removing when I normalize? What are some actual biological things that I'm removing? So I'm pretty sure. Yeah, you're filtering. Perfect. I'm pretty sure everyone here might be a biologist, but I was in a wet lab before, and one key thing is, okay, the data that doesn't fit in the model. Perfect. So yeah, technical noise. So when you do like qPCRs, or if you do certain experiments, yes, blank samples, perfect. You're keeping everything to a bar. So you might have a certain threshold. So if you use a blank sample, that might be your threshold. So yeah, you're getting rid of all that technical noise that maybe you might have added extra reagents into your experiment that might have, caused an increase in that particular sample. So it's good at the end of the day, human error, yes. I'm pretty sure all you biologists out there have been through that. Yeah, you definitely have, some human error in every experiment, and these key tools reduce that error. So quantile normalization helps. Yeah, outliers too. Yeah, because maybe you did an experiment, and let's say it was 11 p.m. at night, and you have to go home and cook dinner for your family or something, and then you accidentally added too much running buffer to your SDS page gel or something or whatever. I'm just giving you an example. And
Yeah.
so
that doesn't make any difference for the buffering SDS page.
I know that's a terrible example. I know because that's actually
consider buffer, but I would consider sample from different sample. That would really mess up.
Yeah. Like if you mix two samples together, yeah.
You run it from one well to another, yeah, that would really mess up.
Yeah, I've done that before. It's not fun seeing extra bands. But yeah, definitely.
Kind of point out on the outliners. If I don't want to get rid of, because there might be some kind of data there.
So
sometimes
I guess
outliners gives you an idea that something is going on that could not be due to the human error or anything. It could somehow indicate something else. Can you not always get rid of them?
So I guess that would be on your threshold. So if depending on what you're looking for, if you set your own threshold per experiment. So if you choose a value of, I don't know, 5, and you see an outlier and you think, you know, maybe that is a significant result. So maybe you might decrease your threshold to 4. You know, so you have a point, and that comes down to deciding your threshold. You know, how low do you want to make your bar when you normalize data? You know, how high do you want to make it?
Sorry, maybe I could just add to this. So the threshold is actually for normalization to happen under the threshold. So typically the outliers that we care about are outliers to the top of the scale, not to the bottom, because at the bottom you have very noisy data as represented here. Essentially noisy means that there's not significant difference between conditions, and even if there is, it's below the point of detection. But on the top, for example, if you have very highly expressed genes, that seems to be likely significant. So the threshold that you select here, everything below the threshold gets normalized. Everything above the threshold is going to stay the same.
Yep. Perfect. Thank you for adding in. It's a good explanation. Perfect. So let's go on to the next slide. So yeah, just as Ilya said, so over here in this particular set of data, before you normalize it, you might feel like the first column represents something, but after you normalize it, they all seem to be at around the same range. So there might not be a true signal based on it, you know, if you don't normalize your data. So that's the take-home message of the normalization. So this is just a scatter plot that represents, you know, how normalization can help you compare different points. Most of the points here are at the corner of your graph, and you can't really come up with a proper correlation. But after you normalize your data, you can see most of the distribution in the center of the graph with a correlation line, and you can come up with better inferences on whether or not you should continue your experiment or perform other types of steps. So just some code to review of some code from previous lessons. So as you can see here, you read the table with the read.table function, and you provide your link to wherever your data is stored. And then you index for a particular, you know, or you get rid of whatever you don't need in your particular table. So in this section, you're removing column 1, and then you want to create colors for your box plot. So you create a vector, a character vector here, or a C vector, and then you reference it to the colors variable right here. So colors, and then you have the rep function. So rep is replicate. So you give it whatever you want to replicate. So red, and you 6 times, because there's 6 samples here that represent luminal. So you replicate that 6 times, and same with blue, 3 times. So at the end of this, you have a whole vector full of 6 plus 3 plus 2 plus 2. So that's around 2, 4. That's like 13 words in this character vector. And then when you use the boxplot function, you specify the data you have initially. So you put the data in this df variable, and then you have a character vector that represents all the colors you want to name your your column. So the first column will be red up to 6, because it's the character vector says red, 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 red 6 times, and then it's blue, 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 then green, green, then gray, gray. So this is just some review of some code that we did earlier. And if you have questions, just type it in the chat and I'll get to you. And so just some review, we did a previous assignment, and you had to create visualizations with some Excel data, and then you also had to load some data from this the 15 gene data set of breast cancer data set, and then you had to visualize gene 10 as a bar plot, and then answer this important question. So this is testing your analytical abilities of whether you can create, a conclusion based on the data or the visualizations that you've created. So we'll go over some syntax. So can someone tell me what this does? Read Excel. So this function here, read -Excel. Yes, okay, perfect. Read an Excel file, perfect. And how about the sheet equals to? What does that mean? As it says, comma, sheet equals to.
You specify the sheet that you want to read?
Perfect, yep. Perfect, perfect. So then, after you read okay, yeah, so perfect, great. So you reviewed this data before, and you created a table, and you summed the categorical data, you found the percentages, and then you created a bar plot and specified margins, and you had a title. Perfect. So then you have this bar plot here, and it represents that this bar plot says gene 10 plays a role in luminal breast cancer because all the luminal samples are high, if you notice that. And then for this particular bar plot, gene 6 is high in every sample except the claudin low. So that means gene 6 might not play a role in claudin low. And finally, in gene 2, gene 2 appears to be a gene that's in normal-like breast cancer. So you see normal here. So that is definitely, it's probably related to a normal function because it's not present in luminal breast cancer or basal or claudin low. So it's probably something favorable to have. And so, as you can see in this scatter plot, so can someone tell me the difference between the top scatter plot and the bottom? What's the difference between the two? Yeah, perfect. Lydia got the point. Yeah, the top one has correlation because there's a diagonal, the points are more closer to the diagonal, perfect, while the bottom one doesn't. So you're comparing normal breast cancer to normal, and there appears to be a correlation in the scatter plot. Perfect. And then this is another type of metric. So you have scatter plots, you have line graphs here, and correlation coefficients. So correlation coefficients, if the higher the number, the more likely the correlation. For these graphs, if you see 2 bumps, are like that usually signifies better correlation. Scatter plots, if it's more diagonal, just like Lydia mentioned, that there's correlation. So perfect. And this is another assignment that you send the code you showed us before. Yeah, I'm pretty sure we can do that. Maybe someone someone in our team will do that with the email. Yeah, 100%. I'm pretty sure we can do that. All right, and then yeah, we have to work on this assignment. So make sure you're gonna start a pipeline for gene expression in one of the modules, and we'll get to it. I'm pretty sure you started part of it in transcriptomics, and I think that, and you have to do assignment 2, which
And
is
well, can I just
Go
briefly
ahead.
mention the code that you showed, the graph that shows the scatter plot and the density plot and the correlation values together? So I believe the
Oh,
question
yeah,
was,
I can
what is the code to produce
this
this
one.
table? So this is a specific package in R called ggpairs, and it relates to ggplot in general. So this is covered, an introduction to ggplot visualization is covered in the advanced visualization in R course or lesson. But this is a specific package that allows you to combine multiple plots. And so when we talk in the context of exploratory data analysis, instead of just plotting one plot at a time, like a box plot or a scatter plot, you can do multiple comparisons at a time. And so here you see a scatter plot between pairs of samples or pairs of types of samples. And you can also see a density plot and a correlation value. You can actually add more to this. You can do box plots and scatter plots, or box plots and density plots. And so we will post on the forum a little link to ggpairs, which is a specific package to do this kind of analysis.
Awesome. So yeah, that package is useful. I've used it in my coursework and research, so it is helpful. Perfect. So yeah, so make sure there are 2 assignments that you have to work on. I believe they're in transcriptomics 1, and you have to load a full gene expression table and compare these these types of cancers, and you have to find which sample you have to characterize the samples in your data set based on the visual that you create. And I think we'll go to the coding part. So does anyone have any questions so far? So, okay, if not, I'll jump to the coding portion. So yeah, so make sure after this session you do transcriptomics 1 and work on that video. So, and those assignments. So make sure you join that. 1 second. Maybe, yes. You need to complete transcriptomics 1 after this. So yeah, Beepsa said that she will email the assignments and codes with the videos. So yeah, we're not done yet. I just wanted to say that part before. So I finished the PowerPoint. So now we're going to do the R lesson right now. So yeah, you can watch the video later, but we'll jump to the actual lesson here. I hope everyone opened their courses to this advanced data visualization portion. So we're starting in histograms and density plots. If everyone jump to that section. So just give me I don't know, say something biologically related if you have opened this section. Or you can just say, yes, I've opened it. Is everyone here? All right, okay. Perfect. Okay, so let's start off with this review. So we made So to make a histogram, you need to convert your data into a matrix form. So let's say you have a data frame, so called log df. So you have to use this function called as matrix, which forces your data frame to convert into a matrix of data. So it removes like the columns, the column names, and it just keeps numerical values. And then once you have that numerical matrix, you use the histogram function, HIST. So there are certain parameters that you put into the histogram function. So you put in the actual data set that you'll use, which is the matrix of values, where we are, 0.2. We are in 0.4. So I hope, do you guys need help with the first 3 points, or should I just, like, are you guys fine with the first 3? So go to advanced data visualization if you haven't, and jump to 0.4. Are you fine with 0.2, Lydia, or are you stuck somewhere? We'll get back to you if you're stuck on 0.2. Okay, you're not stuck. Perfect. Great. All right. So back to this. So when you write main equals, that's a generic title function for most of your graphs. So over here, histogram for all samples. Then you write, you write whatever title you want. You can say, yeah, histogram for all samples. So main equals that. Can someone tell me what xlab means? Okay, axes. Which one? Yes, perfect. Perfect, the x-axis. So xlab will be the label for your x-axis. So it's weird. Why isn't it here? But that's weird. The value, it just says value here, but it should actually say xlab. But then breaks. Can someone tell me what breaks means? Do you know what the word breaks means? Yes, divisions. Yes, bins. Yep. I'm pretty sure most, like when you deal with experimental data, you have to, break them into intervals. So those breaks, yeah, or bins. So you break your data. Yes, perfect. And then over here, it's the color scheme. So rainbow is a generic color function, and it chose 11 different colors from rainbow. So then it labels each of your histogram samples with a different color. And then you have the frequency and then the value on the bottom. Perfect. So this is just another histogram on the bottom over here, but just for one particular sample. So again, whenever you make histograms, you need numerical data. And in order to do that, you have to use the as matrix function. So it forces your data into just purely numbers. And then you use the hist function and you put non-malignant, or like the data set you're using. And in this case, we didn't use rainbow as previously. We just chose the color blue. So over here, the color is just blue, and it shows you your different samples in the histogram. So for this sample, you chose a different, so in the previous one, you chose 1, so that's a non-malignant sample. But in this one, you chose 6, so that column represents Claude and Low. So you select that data with that index, and you use as matrix to convert it into numbers, and then you create a histogram, just like the previous example. Why there is 1? Okay, so the 1 represents the number of the column. So let's say yes, it is the column. It is column, yes. So 1 is the first column of the big data frame. So the first column is non-malignant numbers. So you chose the first column. So all the numbers related to non-malignant. So, and then over here, you chose the 6th column, which is all the numbers related to Claude and Low. So each column represents a breast cancer type, and each row represents the gene or the values associated with that breast cancer type. All right, perfect. So over here, in this particular example, you want to overlay histograms. So sometimes it's easier to compare 2 different samples if you put the histograms on top of each other. So over here, you extract the required columns. So you can also extract with the comma. So log the name of the data frame, and then you put comma and then 1. So that will extract the column and all the row values related to that column. And over here, you'll do the same thing. So you have 2 samples extracted in 2 different variables. And then you have to describe a color scheme. So when you overlay 2 different histograms, you want to have some sort of transparency, because if the colors are too dark, you can't really tell the difference between the histogram on top and the histogram on the bottom. So there's this function called RGB, and each one of these numbers represents a color. So red, green, and blue. So this particular color is red, green, blue. So this one's blue, 1 is blue, and the number here is the transparency. So this is 50% transparent. So the histogram is 50% transparent. And same one here. You chose a different color, so you chose red here. So this one's blue, this one's red. 2 different color schemes. So then you come up, you draw the first histogram using the techniques that we learned earlier. In this particular example, the bins are different, the breaks are different. You don't put an X label or an X label here. You come up with a title, and you put the first color scheme in the first histogram function. For the second histogram function, you have to add this function. You have to add this particular code block called add equals true. So this is the reason why it overlays. If you put add equals true or t, this histogram will overlay on top of this histogram. And again, you don't specify the titles for the second one, because I believe they'll overlap, and you don't want to have a messy histogram. So you don't need a second title if you've already specified one. And perfect. So then afterwards you can see both histograms. So add, essentially, if you put that there, it overlays the 2. I believe if you don't put it there, you'll have 2 separate histograms. So you plot the first one, and then if you write add, then you're forcing this histogram to go on top of that one. But if you don't put add, you'll have 2 separate histograms. And sometimes it's easier to compare if you overlay the histograms on top of each other. Does that make sense? I hope that clarifies the point. If not, just ask me a follow-up question. So adds just telling the second histogram to go on top of the first one. You make it different colors and compare. Yeah, you could also do, yeah, that's also a possibility. If there are two ways of doing it. One way is to create two histograms and overlay it. Another way is to come up with different colors. Yeah, there are different ways of representing data. Yeah, good point. So go ahead.
If you do the add on the, does it matter to do the add kind of on the second histogram code or on the first one? Does it matter?
Maybe okay. I believe you have to put it on the second because you need to put it on. You need to overlay it on something else. Otherwise, but if anyone has a like Ilya or anyone has a comment on that, because I believe that's how it is. You need to put it on the second one to overlay on the first one.
Understood. Thank you.
Perfect. So let's go to the next portion here. So over here, we're creating density plots. So they're kind of like cleaner histograms. So they just they're smoother. You know, they're curved instead of seeing all the bars. And for essentially, if you want to create these histograms, or I mean these density plots, there's a particular function called density. And as we defined here, we collected sample 1 from the previous code over here. And you use this density function to convert that data into a density plot type. And you just plot that object with the plot function. So plot. So the density, is it's a function in R that converts your data into a density plot. So I believe it's an object. I don't know if you've learned about those particular objects yet, but we'll go over when we do the exercise. I think it will make more sense if you're stuck with the function, because I believe when you do it, so density plots are like the histograms, but more smoother. So it's describing the same type of data, but it looks crisp. So like over here, you see this this type of curve, and there's like a similar type of curve here. You see how there's this red bump up here, and there's this kind of red bump up here. It's the same trend, but it looks more appealing to the eye. It's smoother. Does that make sense? Like it's the same trend, but instead of bars, you create like a curve. That's the only difference, or that's really the main difference between a density plot and a histogram. It tells you the same type of data. I hope that clarifies. Yes. Yeah, yes. The same, just like we compared the two samples in this histogram, you compared the same two samples in a density plot, but it looks, you might even say it's more easier to compare, but that's depends on what visual you like. Yep. So it's the same two samples. Does that make sense? I hope I clarified your point there. If not, just keep throwing questions at me. Perfect. All right, so let's keep going. So again, so you create the density object, and then you click plot, and you specify a color, and you create the shape. So you put your particular, you define the shape, and you have your border color for the shape. So over here it's the same color, so color 2 that we specified earlier. So color 2, if you forgot, it's right here, it's red. So red, so the, okay, perfect. Let me, yeah, the question. Okay, sorry, perfect. So then we have a heat map here that represents, you have to, if you want to compare genes, you convert it into a matrix, and then you put it as a heat map to compare different values of, so you can see easier trends on a heat map. And elaborate on polygon. So polygon is essentially the type of shape that's on the density map. So it's a function that specifies the color borders. So you mentioned the border of the color, and you see this line, and then everything's just the same color. So red, and the inside, you fill the inside red, and you use the same data. So it's just specifying the way the colors that the curve represents. It's not very it's not as difficult as it sounds, because polygon's just a random function here. Like, it just sounds random, but it's not difficult. Yeah, it's a plotting function, really. It's don't stress too much on it. When we do the assignment, it will make more sense. So perfect. So over here in the heat map, you convert your data into a matrix, and you use the heat map function, and you specify your column size with CEX col. So yes. So now let's practice, because I think the best way to learn is by practice. So I hope you. One thing I forgot to mention is that heat maps are very good at visualizing differences between gene expression data. It's easy to compare 20 genes to each other, or different sets of genes to each other. So it's kind of a common visual used in science to represent differences in gene expression. So heat maps are a very valuable tool that you'll learn right now. So let's do that right now. So I hope you jump to the practice portion. And yeah, take your time to do the practice. And I've done it. Try to do it on your own. And if you have questions, let me know in chat. Essentially, it's the same coding blocks from earlier. Yep, you have to create a density plot, histograms.
So just to clarify, the reason why we want to look at these different types of visualization is because we have a way to plot one single variable, for example, gene across samples, or we can combine all the data in our samples. For example, all the genes together, you can visualize as a density plot or a histogram. But a lot of times when you look at everything together, you don't see individual trends. For example, in box plots or in histograms, you can assess the overall structure of your data. For example, how distribution of data looks, or how skewed it is maybe, or something like that. But in individual bar plots, if you just look at a bunch of different bar plots, it's hard to compare them one to the other. So heat maps kind of combine that information together. They show us individual gene patterns, but across multiple genes. And now when we look at the heat map, what we also have is the ability to organize them so that the ones that are closely related are closer to each other. And if you have ever tried to do something like this manually, it's actually a very tedious process, because you take two and you compare them, and then you move two closer together, and then you take another two, and again, you can reorganize their order. And here what we have is an ability to automate this process. So the heat map is very useful in that sense. And it's also a good introduction to clustering, which we will do in our next part of this program.
All right. So I hope if I hope you understood those exercises. Oh, okay. So maybe I'll just you know, I'll show you this heat map function here. So we have a nice heat map over here, and you can play around with your heat map by adding different. You know, you can change the size here. Maybe that might be too big. Let's see. See, now you can't see the bottom axes anymore because it's too big. So, and there are other parameters on the heat map function. Like you can change the color, and the you can specify different lines. Like if you want to, like if you want to divide, if you want to put a line in between to divide cancerous versus non-cancerous. So let me just pull up some parameters so I can play around. So let me change some colors here, because it starts to get very tedious when you add, like, the ggplot package. It's great, but they're also you'll find out there are a lot of parameters that you can play around with. So it's good to have some sort of documentation. So let me play around with some colors. Okay, let's see what. Pretty sure it's color. So maybe I want to add, I believe this should be it. If not, I will double check. Will that change it? Well, this doesn't help us, but but if if you divide it into different colors, you need to create a proper color metric like earlier with rep. So you definitely need to create better better divisions of your data. Let's see what other parameters can I really change here. Any suggestions?
Well, so you can actually change whether you do clustering on both genes or columns and rows, or you don't. You have the option to, for example, do a different scale. So there are multiple options in the heat map, and this is a basic heat map, but there are some also more advanced heat maps that you can look into later. And we'll take a look at that when we do differential gene expression.
Yeah. Yeah, when you get to that, it will definitely, you'll enjoy making these colorful visuals. Like I've had to make them for my different projects, and they're very fun to do. I have a bar plot log 2. So when you come up, when you have an error like that, let me teach you some self debugging tools, like, so you can also figure out how to solve the error. So I would first, I'd maybe start with 1 to 20 and see what that prints out, to kind of give you an idea of what you're doing. So wait, let me comment. Sorry. Oops.
So, Clinton, for the sake of time, again, this is, we're already an hour and a half into the session. And one of the main things, what we wanted to achieve today is also get to Python, which I don't believe anyone had a chance to do just yet. So maybe you could switch over to that.
Yeah. So let's go to Python. So, okay, we'll learn the code here. So Python is, it's similar in some ways to R, but it has its own purposes. You can make games with it. You can, do more machine learning and artificial intelligence applications. So it's, when you first start off with your code, you have to import your packages that you require. So in this particular case, you import a package called pandas and a package called numpy. So this is the basic syntax when you start your Python program. And then, because we're working with biological data, normally we have to import our data. We need to take it from somewhere. So it's similar to R, but instead you write pd.readTable. So this particular function, you provide the index of where your file is located, and you provide a separator. This represents tabs. So your data is separated by tabs, and that just organizes everything into columns. So if instead of tabs, if it was comma, then it would separate it based on comma. So it would divide your data into columns based on the comma. And then this header portion just gets rid of the, it depends on how you set it. So if you have header equals 1 or header equals 0, it has certain column names. So it sets the first column to, it sets the first row as the column names of the particular data frame that you're pulling out. So this is how you read a file into Python. And you can read different types of files. So this is just a text file. So maybe you want to read a Excel file, so XLS. So you just copy the same link, but but just a different file name. But this, since it's XLS, you don't really need a separator because it's already arranged into columns and rows. But this particular file is a CSV file, and so depending on your CSV file, you might need a separator. So this one does. So it has a semicolon to separate your data values into columns. And then if you just want a rough view of the type of data you have, when you reference your data sets and you put it into the df variable, you just have to write df.datatypes, and it tells you whether you have string data, whether you have number data. It tells you the type of data you're working with. Can we work with just nucleotides? Yes, it depends. Like there are certain Python packages that allow you to like take, like instead of this being a .csv file, it might be a .fa file or a .fasta file. So it will read your sequence and help you separate it into, like, it will get rid of the first part of the sequence, you know, the greater than sign and the numbers, and you know, maybe the quality scores and all that. So it will, there are packages that help you.
Clinton,
Go ahead.
okay. So I would like to add to this. So there is package like BioString. So they actually handle those sequencing data, nucleotide sequencing data. So this is one of the packages that you can export. So I think that would be quite useful.
Yeah. I think
So we have actually one exercise even on that for the how you can read a sequencing data, but that is not under here. So that is under the, I think, genomics courses, as well as introduction to the bioinformatics, where you can actually how you can read this particular data, nucleotide or of amino acid. Yeah.
Yeah. So when you say qualitative data, what do you mean? Like, do you mean like
Non-numeric, I guess, is it?
yeah, true, but is there a particular type of data you're talking about, or just okay? No, yeah, it can read, like it can separate the name of your FASTA file into different rows. So let's say you have a big file with different chromosomes and you want to make that into a nice data frame. So there are packages that will, you know, help you sort the sequences, and then you will also have the chromosome ID right beside it in its own row. So you might have the sequence and then a column for the sequences and a column for the chromosome number. So yeah, you can definitely you can just, you know, pull out those types of data, and if not, we'll teach you the skills to do that on your own in addition to the packages, because everyone's doing novel research. I believe she mentioned the bioinformatics course, the string package, for example, that will introduce you into that topic, you know, because yeah, a lot of scientists need to, you know, do that type of data. All right. So anyway, but yeah, you can definitely follow up with us if you have more questions about that. We're running a little bit low on time, so let's just do some of these exercises. So if you could try some of these exercises on your own, and then hopefully we'll, you know, go to the concluding remarks after this. So could you try these exercises on your own? So I'll do this one as you do your own. So click. So I have to. Go ahead. You have a question? Which course, Harpreet, could you mention the course that Lydia is interested in?
So that is introduction to the bioinformatics as well as under genomics.
Okay. Okay. So I will leave you with these examples. If anyone wants to.
So I think, Clinton, you can show with one example.
Okay, I'll show one example. Perfect. All right, so let's say I want to import this text file. So, well, I guess you
So
copy.
first, okay, you have to download this file that is available under this link. Then you have to drop that file under this drop a file option. But before running this code, you have to load these codes. I mean, load these packages, import these packages. So we have both the in R as well as Python. Okay, so
yeah, I think so I need to do this one. So I need to drop the file here.
okay, so you have to drop a file. I mean, you have downloaded that file.
Yeah.
So you just need to drop that file. I mean, drag that file.
Yeah, I have to.
Yeah.
It's weird. Or sorry.
Save on your system.
Yeah.
So now drag that file to the drop a file option.
Oops, I think, sorry, I might have not saved it in the correct location. Is it, oh, it's there, but this one. Oh, there it is. Perfect. So pull that, drop it here, and then click run.
No. So
Oh,
before
sorry.
that, you have to, import those.
Packages,
Okay,
my
so
bad. I did
can
not.
you go over the first step?
All right. So, yeah.
Yeah. First, you have to call these packages. So once your packages get loaded, now drop that file to the first Dropbox.
Oh, this one, eh?
Drop a file option. Yeah. So just drag your file to this box. Yes. So now you can run this code.
Perfect. Yep. So now it pops up. You see the data set that you're working with, and it's separated. Yep, you can see the space into these columns. So yeah,
Yes, Lydia. Sorry to interrupt. Yes, that is course for introduction to the Python. Not introduction to Python. I have to check that whether we have that lesson there. That lesson might not be available under this introduction to the genomics. So that is introduction to the bioinformatics. There is other course.
All right. So I think if does anyone have any? Does Ilya, do you want to take over from here? I guess.
So
they can do.
I mean, okay. So Ilya has already left for the other session.
Okay.
So if you have any other question, so you can post your questions in chat. I mean that at forum as well, and you can email me as well. That is at sport at. Let me just at Pine Bio. So you can email me your questions at this mail ID. But if you want to discuss with everyone, you can post directly in the forum as well.
Yeah. Yeah. And yeah, I guess.
So in the next session, we are going to cover exactly how you can also visualize your data in the Python as well. So thank you everyone for joining today's session.
Thank you for joining. And yeah, we'll definitely do more creative things. My, biology is fun.